Food safety has become an increasingly critical concern in today's globalized world, where food supply chains are more complex and interconnected than ever before. Contaminants, pathogens, and chemical residues can compromise the safety of food products, posing severe health risks to consumers. According to the World Health Organization, foodborne illnesses affect approximately 600 million people globally each year, leading to 420,000 deaths, with children under 5 accounting for nearly 30% of fatalities. In response to this growing challenge, scientists and researchers are developing innovative technologies that offer more rapid, sensitive, and accurate methods of detecting contaminants in food products. Among these technological advancements, quantum dots have emerged as a revolutionary tool with the potential to transform food safety detection systems. Quantum dots, which are nanoscale semiconductor particles, exhibit unique optical and electronic properties that make them highly suitable for sensing applications. Their ability to fluoresce at different wavelengths based on size and composition enables highly specific detection mechanisms that traditional methods cannot match. This documentary explores the underlying science of quantum dots, their unique advantages, and their groundbreaking applications in rapid food safety detection. Quantum dots are typically nanocrystals composed of semiconductor materials such as cadmium selenide, cadmium telluride, or indium phosphide. These nanocrystals range in size from 2 to 10 nanometers, roughly equivalent to 10 to 50 atoms in diameter. Their small size results in quantum confinement effects, which means the physical dimensions of the particles directly influence their electronic and optical properties. As a result, quantum dots exhibit size-dependent fluorescence, where smaller dots emit light in the blue spectrum and larger ones in the red spectrum. This tunable fluorescence is one of the most critical features for detection purposes. When exposed to a specific wavelength of light, quantum dots absorb energy and re-emit it at a longer wavelength, a phenomenon known as photoluminescence. These emissions are exceptionally bright, stable, and resistant to photobleaching, making quantum dots ideal candidates for developing highly sensitive biosensors for food safety applications. Furthermore, the surface chemistry of quantum dots can be modified to attach various biological molecules such as antibodies, aptamers, or peptides. These functionalized quantum dots can then be engineered to bind selectively to specific pathogens, toxins, or chemical contaminants present in food, making them versatile tools for targeted detection. The modern food industry faces mounting pressure to ensure food safety in response to increasingly stringent regulations, consumer demand for transparency, and global trade requirements. Traditional food safety detection methods such as enzyme-linked immunosorbent assays, polymerase chain reaction, and microbial culture techniques, although effective, often involve lengthy processing times, labor-intensive procedures, and high costs. For example, microbial culture tests for detecting bacterial contamination typically require incubation periods ranging from 24 to 72 hours to yield results. In contrast, enzyme-linked immunosorbent assays and polymerase chain reaction methods, while faster, still require specialized equipment and skilled personnel, limiting their use in on-site testing scenarios. These limitations highlight the urgent need for rapid, cost-effective, and portable detection systems that can deliver real-time results with high accuracy. Quantum dots present a promising solution to these challenges by enabling faster detection times without compromising sensitivity or specificity. Their integration into biosensing platforms facilitates the development of portable devices capable of conducting on-site testing in various environments, from food processing facilities to retail outlets and even consumers' homes. Quantum dot-based detection systems operate on the principle of fluorescence resonance energy transfer, or FRET, or direct photoluminescence. In a typical quantum dot-based biosensor, the nanocrystals are conjugated with biomolecules that selectively recognize and bind to target contaminants. Upon interaction with the target analyte, changes in the fluorescence signal, either an increase or decrease in intensity, indicate the presence of the contaminant. One practical example involves using quantum dots conjugated with antibodies to detect bacterial pathogens such as Escherichia coli or Salmonella in food samples. When these functionalized quantum dots bind to bacterial antigens, the resulting fluorescence change can be measured using a portable fluorometer, delivering rapid results in under an hour. This method demonstrates sensitivity at levels as low as 10 colony-forming units per milliliter, outperforming conventional detection techniques. Another significant application of quantum dots is in detecting chemical contaminants, such as pesticide residues or heavy metals. For instance, researchers have developed quantum dot-based sensors capable of detecting organophosphate pesticides in fruits and vegetables. These sensors use quantum dots conjugated with enzymes that react with the pesticide molecules, triggering a measurable fluorescence response proportional to the contaminant concentration. Recent years have witnessed remarkable progress in the application of quantum dots for food safety detection, driven by advances in nanotechnology, material science, and bioengineering. Several innovations have emerged, enhancing the sensitivity, specificity, and usability of quantum dot-based sensors. One significant development involves the use of Graphene-Quantum Dot Hybrids. Graphene, known for its excellent electrical conductivity and mechanical strength, enhances the performance of quantum dots by facilitating better electron transfer and improving signal detection. This hybrid material has been used to create highly sensitive sensors capable of detecting trace levels of contaminants, including toxins such as aflatoxins produced by fungi in grains and nuts. Another breakthrough involves the development of multicolor quantum dot sensors that can simultaneously detect multiple contaminants in a single test. By utilizing quantum dots of different sizes and emission wavelengths, researchers have created multiplex detection platforms capable of identifying various pathogens, chemical residues, and toxins concurrently. This multi-analyte detection capability significantly reduces testing time and costs while providing comprehensive safety assessments. Moreover, innovations in portable detection devices have made quantum dot-based biosensors more accessible for field use. Handheld fluorescence readers integrated with smartphone applications are now being developed to enable real-time data analysis and remote monitoring. Such technologies are particularly valuable in low-resource settings where laboratory infrastructure is limited. Several case studies illustrate the successful application of quantum dots in enhancing food safety detection across different sectors of the food industry. One notable example is a study conducted by researchers at the University of Illinois, who developed a quantum dot-based sensor for detecting Listeria monocytogenes in processed meats. Their biosensor demonstrated detection limits as low as 1 CFU per milliliter within 30 minutes, offering a faster and more sensitive alternative to conventional culture methods. In another study, scientists from the Indian Institute of Technology developed a quantum dot-based biosensor for detecting pesticide residues in tea leaves. The sensor exhibited high sensitivity and selectivity, with detection limits below the maximum residue levels established by international food safety standards. Additionally, a collaborative project between researchers from China and the United States focused on developing quantum dot sensors for heavy metal detection in seafood. The study demonstrated the ability to detect mercury and lead ions in fish samples with high accuracy and in real time, addressing growing concerns over seafood contamination due to industrial pollution. Despite their numerous advantages, the widespread adoption of quantum dot-based food safety detection systems faces several challenges and limitations. One major concern involves the toxicity of certain quantum dot materials, particularly those containing heavy metals such as cadmium. While encapsulating quantum dots with protective coatings can mitigate their toxicity, regulatory agencies, including the U.S. Food and Drug Administration and the European Food Safety Authority, require thorough safety evaluations before approving their use in food-related applications. Another challenge is the cost of large-scale quantum dot production. Although the synthesis of quantum dots has become more efficient in recent years, producing high-quality nanocrystals with consistent optical properties remains expensive. This cost factor can hinder the commercialization of quantum dot-based detection devices, especially for small and medium-sized enterprises in the food industry. Furthermore, issues related to sensor stability, reproducibility, and shelf life must be addressed to ensure reliable performance under various environmental conditions. Research is ongoing to develop more robust quantum dot formulations that can withstand temperature fluctuations, humidity, and other factors commonly encountered in food processing and storage environments. The future of quantum dots in rapid food safety detection appears promising, with ongoing research focused on overcoming existing limitations and expanding their applications. Several emerging trends are shaping the future landscape of this technology. One such trend is the development of non-toxic quantum dots made from environmentally friendly materials, such as carbon or silicon. These alternative nanomaterials offer similar optical properties without the health and environmental risks associated with heavy metal-based quantum dots. Researchers are also exploring the potential of Perovskite quantum dots, known for their superior photoluminescence and tunability in biosensing applications. Artificial intelligence, or AI, and machine learning are also expected to play a crucial role in advancing quantum dot-based detection systems. In addition, the rise of the Internet of Things, or IoT, is facilitating the development of smart food safety monitoring systems. Quantum dot sensors embedded in packaging materials or integrated into food storage facilities can provide real-time, continuous monitoring of food quality parameters, such as microbial contamination, temperature, and spoilage indicators. These IoT-enabled systems can alert stakeholders to potential safety issues, allowing for timely interventions and reducing food waste. The integration of quantum dots into rapid food safety detection systems represents a transformative advancement in ensuring the safety and quality of food products across global supply chains. Their unique optical properties, high sensitivity, and potential for real-time monitoring position them as a valuable tool for addressing the growing challenges of food contamination and safety assurance. While challenges related to toxicity, cost, and sensor stability must be addressed, ongoing research and technological innovations are paving the way for the broader adoption of quantum dot-based detection methods. The convergence of nanotechnology, artificial intelligence, and IoT promises to revolutionize food safety monitoring, providing faster, more accurate, and cost-effective solutions for industry stakeholders and consumers alike. As the global population continues to grow and food systems become increasingly complex, ensuring the safety and integrity of food products will remain a top priority. Quantum dots offer a beacon of hope in this endeavor, representing not just a scientific innovation but a vital tool in safeguarding public health and enhancing global food security for future generations.